Okay, I think this is my question. So it is actually the plasma membrane is not visible under light microscope, but it's visible under electron microscope. So like in the picture, you probably can see like a border sort of thing, but like that's actually not the membrane you're seeing. You're probably just seeing like kind of this stain of like the content. Yeah, so like if this question said electron microscope, it would be right.
Okay, excellent job, guys. So yes, if you remember the week 1 presentation, that the definition of health according to WHO is a state of complete physical, mental, and social well-being with the absence of disease and infirmity. But the main issue was, main issues are that no one can achieve complete health. There's always going to be some aspect of our health that's not in perfect balance. And the other point is who defines well-being. I know I put in that green answer just to throw you off there. Yeah, it's not about the fact that World Health Organization defines, it's just that it's completely subjective. No one's defining it, it's very subjective. So it's a very subjective definition, thus not a great one. Okay, that's it. Great job, Jay. Nice job, everyone. Yep, so avoidable inequality, as the term suggests, is something unfair that can actually be avoided. So such as your location, because you can move your location, you can change your location. The other three are all unchangeable factors about yourself. That's all there is to it. Ooh, Sammy's just beaten Jade. Good job. Okay, good job. So I thought this would be a tough one, but you guys have done a great job of it. So does someone who selected green want to actually explain why they did, if it wasn't a guess? Why would average body weight of a population not be a health indicator? I'm curious to know what you guys say.
Because it doesn't take into account gender, like height and other things that could affect it.
Yep, that's definitely one of the reasons. So just because you take the average body weight of a population, like it doesn't mean anything, because obviously it depends on, yep, that's right, people's height, people's age. It's very useless. And also, just even if you are like, it has been found that actually that even if you are if you have a healthy BMI, you can still have high cholesterol and other issues, other health issues. So weight is not even the best health indicator. So it's rather not a health indicator at all. Good job, guys. And infant mortality is actually a health indicator, just for those two people. Jay's back on top. Yep, great job. So a bit tricky because I put the blue answer does include some of the people who are eligible for Medicare, but the red answer covers all of them. So Australian citizens and New Zealand citizens in Australia, so not that Medicare applies to people living in New Zealand. Reciprocal country agreement holders, which are select countries that have this agreement that they can use Medicare if they come to Australia, and also permanent visa holders. Good job.
Yep, well done, everyone. So looks like a lot of people selected green one as well. So for option D, instead of membrane protein, it should be membrane lipids. So like, because oh wait, actually, yeah, I guess I phrased it in a way that's kind of confusing. So like my like what I meant was so like, because the lipid was the structural element for the plasma membrane. So and the membrane is selectively permeable. But okay, yeah, I guess this question's phrasing is a bit confusing. Okay, well, because the proteins can also be like channels where like things could pass through. So I guess green one could be right as well. Sorry about that. Yes, well done. So it's great to see no one selected the yellow one or the green one. Phagocytes are not conscious. But so for the first one, basically, the phagocytes, so like the thing that's responsible for the cell shape is actin cytoskeleton, actin filaments. And for microtubules, so although we say they're responsible for cell motility, that is more about kind of cell movement on the surface, for example, through cilia, like in a airway, like cilia kind of just like, yeah, so it's, or like flagella is kind of more about moving the cell through kind of cilia, flagella, that sort of thing. But like this, the phagocyte is more about kind of changing the cell shape. Wow, this is great. Yeah. So cilia is actually found in a lot of parts of the body. So in the bronchus, of course, like for airway, and they're actually in middle ear and in fallopian tube as well. So fallopian tube is a part of the female reproductive system. And for small intestine, so like the thing in that is villi. So that's for digestion. Okay. The question gave it away. Why did it say which microtubule is involved? Well, yeah, central is kind of made of microtubule, and it is involved in spindle formation of during mitosis. Is great that everyone got it right.
Okay, that was my question. But yeah, so where elastic extracellular matrix fibers found? They're mainly found in the arteries. So the picture might have given it away for some people, but that's basically an image of an artery. And why we need it to be, yeah, exactly. And why we need it to be elastic is because, you know, there's different volumes of blood that's pumping through. So the walls need to be able to extend and retract readily. Yeah. Okay, so hyaline cartilage is strong, flexible, and also has resistance to compression, but it's not elastic. The cartilage that is elastic is called elastic cartilage. So that's the feature that is not a part of hyaline cartilage, and all the others are. I think most people got that right, but yeah, so ground substance has glycoproteins, proteoglycans, and also fibers. So you can have collagen or I'm pretty sure it's, yeah, elastic fibers, but there's no blastocytes. That's something that I just made up. Sorry about that. Yeah. Okay, so this is, I think, a very important point, but the Hardy-Weinberg law, it's not true because it's very, what do you call it? It's very hypothetical because there's definitely going to be factors that might influence genes in a population. So there might be migration, mutation, for example. So it's very rare that the law which happen. So yeah, it's likely to be false.
You cut out a little bit there. Is that just me, or is that for everyone? Maybe can you
yeah, she cut out a bit for me as well.
Can you guys hear me now?
Yeah, all good now. So
okay, I'll just go through it again. But basically, the Hardy-Weinberg law is that allele frequencies, they don't change over time in a large randomly mating population. But this is false because in reality, there's definitely going to be factors that influence the alleles in a population. So for example, there can be migration or mutations. But yeah, so the law is false in reality. Okay, there's a bit of an even split here. Oops. Yeah, but basically, what factors alter existing genetic variation? So mutation, however, it doesn't alter what's already there. It brings in new alleles. So that's the answer there. However, for example, we have non-random mating that might increase inbreeding and homozygosity in a population. So it's altering the genes that are already in the population, as well as migration and genetic drift. So the answer there would be mutation. Alrighty, I think most people got this, but yes, a locus is a specific place on a chromosome that's occupied by a gene. So locus kind of sounds like location, and a characteristic of an organism is a trait. Unit of heredity is a gene, and alternative version of the same gene is an allele. Okay, I think everyone pretty much has the hang of this, but yes, catabolic reactions are oxidative. It's a negative delta G, and it increases entropy, and the green box there is a definition of an anabolic reaction. Okay, so the correct answer is entropy. So entropy is a measure of disorder in the system. Energy coupling, it's not the hydrolysis of AT, but it's more so the energy that's released from one reaction that can be used into another reaction. That would be a better definition. And enzymes, oh, you guys didn't pick that. And yep, that's not the definition of metabolism. Yep.
Yeah, so this is false. So a single nucleotide polymorphism, a SNP, is when a single nucleotide, so whether A, T, C, or G in the genome differs between members of a species. So it's not the gene that's different, it's just one nucleotide.
Yep, so uncompetitive enzyme inhibition, that's when a molecule binds away from the active site and it prevents substrate binding. A lot of you picked yellow, which is it binds away from the active site and changes the active site shape. That is non-competitive enzyme inhibition. They sound pretty similar, but yeah, that's the difference. And red is irreversible enzyme inhibition, which is not the case. And green is just competitive because it's binding to the active site.
Yay, well done. A bit of a trick question. So it's two glucose molecules, not one glucose molecule. So for the glycolysis of one glucose molecule will give you 2 ATP and 2 NADH. So for 2 glucose molecules, just do some math.
Yufei, there's also 2 questions in the chat if you wanted to have a quick look. Not too sure if they've been answered. or if we were planning on answering them live.
Yeah, Mahek, do you want to answer them?
Yeah, I can answer them. So the difference between does an uncompetitive prevent substrate release by binding to the active site? So uncompetitive, I believe it doesn't bind. Wait 1 second. Yeah, so uncompetitive, it binds away from the active site. And what's the difference between uncompetitive and non-competitive? I think non-competitive specifically, it changes the shape of the active site, whereas uncompetitive, it just prevents the substrate binding. So that's the main difference between the two.
I think it's actually that the uncompetitive one, it prevents the release, whereas non-competitive prevents the binding. So like non-competitive, they're both going to bind away from the active site. So we call that spot the allosteric site. But the difference is like the time when they, I guess, work, because non-competitive prevents it from binding in the first place by changing the shape of the active site, but uncompetitive just prevents it from being released.
Okay, sorry about that. Thanks, Akanksha.
Yeah, so this question is about anaerobic respiration. So that's about kind of converting pyruvate to lactate or lactic acid. So for the first one, the pyruvate dehydrogenase is a key enzyme in aerobic respiration. So that's converting pyruvate to acetyl-CoA. And for anaerobic respiration, the enzyme is called lactate dehydrogenase. And for blue one, in anaerobic respiration, NADH is actually converted back to NAD+. So that's why it doesn't like generate energy, because NADH is higher in energy than NAD+. So if NAD+ is converted to NADH, that will generate energy. But anaerobic respiration doesn't do that. And without lactate does cause lactic acidosis. So that is the first part of this answer is correct for the yellow one, but it is not treated with sodium chloride solution. It is treated with sodium bicarbonate, which is a weak base. And fourth one is correct.
All right, so this is a little bit split, but good job to the people who chose the blue answer. So if you remember our presentation from week 4, recombinant TPA or tissue plasminogen activator is used mainly to create a protein in the body that is either deficient or non-existent in your body, in the patient's body, and basically that protein can help to fix blood clotting. So by making recombinant TPA, we can fix blood clotting in heart attacks, strokes, pulmonary embolisms, and so on. And in terms of the other answer options, so the red and the yellow ones, they're actually the main uses of PCRs, so polymerase chain reactions, which we also went through in biotechnology. And the green one to create new tissues is what cloning is used for, to repair injured tissues. So just keep that in mind. J is still at the top. All right, great job, guys. So I remember going through this in week 4 as well. So recombinant proteins can make an unlimited commercial supply of human growth hormone. And does someone remember the 2 conditions that human growth hormone is like used for? What kind of therapy? It's really all in the name of the hormone. Okay, in case you don't remember, it's actually used to restore growth in kids who have hypopituitary dwarfism or achondroplasia dwarfism. Yeah, and in terms of the other 3, well, oxytocin is like the love hormone, which is not relevant. Norepinephrine is another hormone which recombinant proteins cannot make an unlimited commercial supply of. And vasopressin is actually an antidiuretic hormone, so it stops you from peeing so much. So yeah, so yellow is the correct answer. Good job to most people. Wait, so before we move on, the 2 types of dwarfism, which can also be found in our week 4 slides, are hypopituitary dwarfism and achondroplasia dwarfism. So this is what HGH, or human growth hormone, can help restore growth. I might type in the chat as well. Yep, so this is a bit of a trick question. 2 answers here. Why is gene editing better than conventional gene adding? So the main official reason is that it allows for internal transcriptional control. Of course, if you edit the genes that are already inside you, then it's more likely that your body will accept them. And for the same or converse reason, if you add genes, then they might be rejected by the immune system because they would interpret it as a foreign body rather than just something that's supposed to help you. So good job to those who picked up its multi-choice.
Wait, sorry. I think both blue and green are right. So like for blue, it is correct that the protons travel from the intermembrane space back into mitochondria through the ATP synthase, which is in the inner membrane of the mitochondria. And green is also correct for some reason. Sorry about that. And a lot of people chose yellow. So it doesn't, so like the ATP synthase doesn't harvest energy from electrons, it harvests energy from protons. So like from the protons that's traveling back through the ATP synthase, and like it harvests the like kinetic energy of that. Yes, well done. So the last one is incorrect because the chylomicron enters the lymphatic system, not the bloodstream, for absorption of fats. And for the blue one, the fatty acid and monoglycerides, they are kind of results of the hydrolysis of fatty acids, and just hydrolysis of dietary fat. It becomes fatty acids and monoglycerides, and they do enter the cell by diffusion. And for the first one, that is correct as well. and for the yellow one is also correct. Yeah, all of them are mindful attitudes. That's it. They're good attitudes. They're mindful. They're nice, wholesome. Yep. So instead of peace of mind, it is presence of mind. So instead of kind of trying to have like peace, which kind of means like not thinking about stuff or like not worrying about anything, just kind of like the cognitive practices would kind of promote just being present and kind of being aware of what your thoughts are, what your worries are. And for the blue one, non-attachment, is also called letting go. So yeah, and that is one of the big four of cognitive practices.
Yep, so the time taken to complete the cell cycle, it's the lowest in bacteria, so they're very fast, usually like half an hour, 20 minutes, and then yeast and mammalian cells. So mammalian cells tend to take about a day or so, much longer. Yeah, I think everyone knows this. Yeah, so metaphase.
Oh, by the way, the acronym PMET is very helpful to remember the stages of mitosis: prophase, metaphase, anaphase, telophase.
Okay, so yep, green is right. So it's basically 2 cycles of mitosis. Yellow is, it's a bit of a trick because it's not diploid, it's haploid. There's 4 haploid daughter cells, and daughter cells have the same number of chromosomes. That's in mitosis, not meiosis. Yeah.
Yeah, so you guys did pretty good on this, but so the answer is yellow, and what it should be is that effective osmoles are non-diffusible solutes. So the fact that they're non-diffusible, so they're not able to go across the membrane, means that water has to move to make the concentrations equal. And normal saline does help treat dehydration, and tonicity is about the non-diffusible. So that's also really important to remember. Yep, perfect. So it does start off isotonic, but then over time it becomes hypertonic and starts going into the red blood cell. Okay, so I'll go through this. So membrane is always, the type of membrane is always really important with osmosis. So that's always something you have to think about, because with osmosis, the solutes aren't diffusible, and it's about the solvent. Solvent moves away from area with impermeable solute. So solvent moves towards the area with the impermeable solute, because again, you want to make the concentrations equal. It is passive transport, so it doesn't require energy. The water just moves. It doesn't just move, but just you can think about it like that. And the membrane is permeable to the solvent and not the solute, hence why the solvent has to move. Hopefully that makes sense, but ask any questions in the chat if you don't understand. Yep, so cortisol is the only one. There's only 3 classes of eicosanoids: the prostaglandins, thromboxanes, and leukotrienes. They all have different purposes, like prostaglandins, and like thromboxanes are for like clotting, and so those are also in our slides as well. Yep, perfect. So malonyl, the formation of malonyl-CoA from acetyl-CoA indicates that it's going to go down the fatty acid pathway, fatty acid synthesis pathway. Yep. So LDL is the main transporter of cholesterol, and this is false. So the more dense the lipoprotein, the more proteins it carries. Yeah, I just filtered around with the wording. So it's mind and body medicine rather than mind and soul. The rest are CAM modalities. Yeah, it's 65%. It's a bit of a mean question, but they do like being mean. So just a heads up. Yeah, all four of these are concerns, and there are a few more concerns as well on top of this. Yeah. Yep, perfect. Yeah, so that's the only situation that you can kind of take control of the decisions. Yeah, so a doctor can withhold information that is under therapeutic privilege. And so it's mainly to do with the information being possibly harmful to a patient, and that's the only reason why you'd probably want to withhold information. Otherwise, obviously, it will impact trust in the medical profession. Yep. So your reputation shouldn't matter in this situation. It's irrelevant. Yeah. Next question.
Yep, so Down syndrome is trisomy of chromosome 21, and XY chromosome makes the person male. So this one is quite important. You need to know that maternal age, specifically over 35 years, is going to be the biggest risk factor for chromosomal disorders. So that's Down syndrome, Patau syndrome, and Edwards syndrome. The other ones that I've listed are more so for other congenital defects, but not really for chromosomal disorders specifically.
Oh, I missed it.
Doing Kahoots gives me
Probably like skip. I reckon skip and then on there. Yeah, so the red one was a reciprocal translocation. The blue was deletion, and the green was just an inversion. Yep, okay, so for this one, I recommend that you use the diagram that you have in your lecture slides, but I've also got that in our PSP notes. So between the epithelial cells and the basal lamina is the basal surface of the cell, and that's why the two are going to be joined by hemidesmosomes. Zonular occludens and zonular adherens are both on the lateral surface of the epithelial cell, and they connect epithelial cells together. And gap junctions are also on the lateral surface of the cells, and they allow the movement of molecules between cells. Yep, so usually the type of epithelial cells that are lining, I guess, tubules like this are simple cuboidal cells. So if you take a look at the image itself as well, you can see that the cells sort of have a cube-like shape. So with the simple squamous cells, they look a bit more like a blob and flatten out. And columnar cells are more elongated and like thin, whereas as you can see, these ones are quite square looking. Yeah, so epithelial cells are actually avascular. This one is again something that I just suggest that you memorize because it's pretty high yield in terms of past exams. So there's 3 requirements for obtaining ATSI status. The first is acceptance by the community, also self-identification and proof of lineage. The things that you don't need to have are knowledge of the Indigenous language or looking a certain type of way. Yeah, so we don't recommend that you use family members as translators because it can't be like trusted how accurate the information they're conveying is, and they might hide information from the patient as well, especially if it's bad news. And then you should be providing Indigenous patients with longer appointment times. So I think My3 recommends booking a double appointment. And it's recommended to always have one Indigenous healthcare worker on the team.
All right, yeah, so most of you got that right. So in terms of what the other three features allow for, the 4 nucleotides allow for the storage of genetic material. The order of the bases being maintained by the sugar phosphate backbone allows for precise copying of the DNA, and then the mutations affecting nitrogenous bases allows for susceptibility of mutation. Awesome. I'm glad everyone got that. Does someone want to tell me why Okazaki fragments only occur in the lagging strand? All right, that's okay, just for the sake of time. So because the DNA strand can only grow from a 5' to 3' direction, Okazaki fragments are necessary in order to allow for that 5' to 3' growth. Cool. So yeah, folding is a post-translational modification, so that occurs after the polypeptide chain has been formed. All of the other ones occur is what occurs to change the pre-mRNA into mRNA. Okay, I feel like you guys have been working on your ethics assignments, so I feel like there isn't really much for me to explain here. So we can move on to the next one. Awesome. You guys have got that all under control.
All right, guys, so it's just hit 4 p.m. now, and I think since we said it's from 3 to 4 p.m., we might have to cut it off there today. But thank you so much for showing up, and yeah, we will be putting up the full Kahoot into our public Kahoot collection, and if you have any questions, please feel free to DM any of us about the relevant question. And other than that, we'll see you next Wednesday, and stay tuned on Facebook for more updates. And subscribe to our YouTube channel as well. See ya.
Coming, guys.
Thanks, everyone.
Thank you.
Bye.